Hi, it's Zach again. For this homework assignment, I'll be discussing question number 3 from chapter 10 regarding the types of pathogens in and the design and safety requirements of a biosafety level 3 lab. So most pathogens and microbes in a BSL-3 lab are those that can cause issues in the human respiratory system. These agents may be either indigenous or exotic, and most often have a high likelihood of aerosol transmission, meaning that these illnesses can be airborne or spread through the air by a cough or a sneeze from an infected individual. Diseases these agents cause can be seriously life threatening and sometimes fatal. Treatment options are sometimes scarce, and many pathogens that are researched in a level 3 lab may still be poorly understood due to their rarity. Agents studied in this setting include Mycobacterium tuberculosis, the bacteria that causes TB in humans, Brucella, which is a zoonotic bacteria that is spread to humans via animal fluids and causes a slew of symptoms. The CDC has actually labeled the Brucella species as weaponizable due to the unspecifiable symptoms and targeted body systems. Another pathogen is the West Nile virus, which, as you can probably guess, causes West Nile fever. Also, human coronaviruses such as SARS, MERS, COVID 19 are studied in BSL-3 labs due to their mode of transmission as well as their infection and mortality rates. Uh, since this level is considered a high containment zone, the goal is to keep pathogens in the lab. So, what's inside should not be able to leave, and since most of these microbes are airborne, these precautions prevent just that. As far as infrastructure goes, the building and/or lab access is strictly controlled and surveilled. In a BSL-3 lab, there are typically two auto shut and locking doors. One is used for entrance and the other for exit. All non-authorized entry is restricted. Only personnel who are well versed, educated, and trained in biosafety with the microbes within the lab in which they're working should have access. Uh, most air supply to the lab cannot be recirculated and must pass through a HEPA filter. Additionally, air pulled into the lab should come from clean surrounding areas and move toward more potentially contaminated areas within the lab. Appropriate PPE must always be worn. Once again, since we're working with aerosol pathogens here, respirators with independent air supply are likely necessary also. Um, in addition to this, all work must be performed in a biosafety cabinet. Street clothes are mostly prohibited, so researchers may change into scrubs to prevent contamination to those. Hand sinks and eye wash stations must be present and accessible in case of an emergency or possible contamination from a spill, needle stick, glass break, or equipment malfunction, both inside the lab and in the ante rooms. Most biosafety lab agencies and facilities are required by law, actually, to keep a log of any incident and report or publish any instances of such. The general purpose of all precautionary measures in a biosafety lab is to protect not only the scientists in the facility, but also, um, probably most importantly, the public from infection. That's it. These are my references. I'll attach them in a separate document. Thanks for listening.